third time's a charm. I'm going to be talking about beta oxidation, otherwise known as fatty acid oxidation, because we're turning a fatty acid and we're breaking it down into acetyl-CoA. So, as I just said, we're taking a fatty acid. We're not turning it into acetyl-CoA just yet. There's a lot of steps before that happens. So the first step is in the cytosol. We're using acetyl-CoA synthetase to turn a fatty acid into acetyl-CoA. That's known as the activation step, is this first step, because ATP is being put in to be used as energy. It's putting energy into the step. So the next step is when CAT1, the rate-limiting enzyme, turns acetyl-CoA into acetyl-carnitine. And it does that by, notice acetyl is the same. So all you do is remove the CoA, stick on a carnitine, and that's how you get here. That occurs in the inner mitochondrial membrane, that step. Now this next step, so we needed acetyl-carnitine because acetyl-carnitine is the only one that can move through the inner mitochondrial membrane to get into the matrix. Now that we're inside the matrix, CAT2 is going to take off carnitine and put back on CoA. So we get acetyl-CoA again. Acetyl-CoA is going to use medium chain dehydrogenase, MCD, or acetyl-CoA dehydrogenase to turn into trans-enoyl-CoA. Trans-enoyl-CoA is going to use enoyl-CoA hydratase to turn into hydroxyacetyl-CoA. Hydroxyacetyl-CoA is going to use hydroxyacetyl-CoA dehydrogenase to turn into ketoacetyl-CoA. And ketoacetyl-CoA is going to use thiolase to turn finally into acetyl-CoA. I remember these 4 reactions by a mnemonic OHOC because this is an oxidation reaction, a hydration reaction, another oxidation reaction, and a cleavage reaction. And I also remember the cofactors for these enzymes. I noticed that dehydrogenases are always paired with either B2 or B3. So we have a dehydrogenase here, so it's B2 here. Dehydrogenase, but it's B3, so it's 2, 3, 5, 5, 5. So in like ascending order going this way. I noticed that. I'll talk about this in a second. But so typically the fatty acid that we're breaking down in our body is palmitic acid, which is a saturated fatty acid, which means it has no double bonds. It's completely saturated with hydrogens. So we put in a 16 carbon molecule and we get a 2 carbon acetyl-CoA each time we go through this process. So 16 divided by 2 is 8. So that's why the products of even chain fatty acids are 8 acetyl-CoAs. And then every time we go through this process, we produce 1 FADH2 and 1 NADH. So if a fatty acid that's 16 carbons goes through this process, it essentially has to go through this process 7 times because 16. So if we go through this process, we'll lose 2 acetyl-CoAs, so we'll have 14. Go through this process again. Now we'll have 12 left. Go through this process again. There'll be 10 left. Go through it again. 8, 6, 4, 2. Essentially, we go through the process 7 times, but there's going to be 2 acetyl-CoAs left over. So I know it's confusing, but you go through this process 7 times. So you get 7 FADH2s, 7 NADHs, and 8 2-carbon acyl-CoAs. Hopefully that didn't confuse anybody. But not all of our fatty acids are even number of carbons. Sometimes they're odd numbers of carbons. So let's say a 17 carbon fatty acid had to go through this process. It'll go through this process until there's 5 carbons left. When there's 5 carbons left, the 5-carbon molecule will become acetyl-CoA, but it'll also become propionyl-CoA. Propionyl-CoA carboxylase will turn propionyl-CoA into methylmalonyl-CoA. Methylmalonyl-CoA mutase will turn methylmalonyl-CoA into succinyl-CoA. And just recognize that carboxylases are always paired with biotin, and I'm not sure if mutases are always paired with B12, but just know that this enzyme, methylmalonyl-CoA mutase, works with B12. Once succinyl-CoA is produced, it can do a few things, depending on what's going on. So succinyl-CoA can continue through the TCA cycle to produce energy. In the product, at the end of the TCA cycle, it can be turned into oxaloacetate. Then oxaloacetate will turn back into pyruvate and then go through gluconeogenesis to produce glucose. Or succinyl-CoA can be used to make heme. Later, we'll learn that heme is made from succinyl-CoA and glycine, an amino acid. So odd chain products. So notice here, even chain, it's 877 if you're working with a 16 carbon fatty acid. And then here for the 17 carbon fatty acid, it's 777, and then 1 succinyl-CoA. Sometimes when fatty acids are longer than 22 carbons, they need to be broken down in the peroxisomes. So it's called peroxisomal beta oxidation. And once the fatty acid is broken down enough, it'll leave the peroxisome and go to the mitochondria to do beta ox. Normally, peroxisomes don't have electron transport chains, so they're not going to be producing as much ATP as when beta oxidation is done in a mitochondria. At the end of peroxisomal beta oxidation, NADH is going to be sent to the cytosol, but FADH2 is going to be used to make peroxide because peroxisomes make peroxide. It's kind of in the name. So some of the tissues that don't do beta ox, which means they don't use fatty acids as energy, are red blood cells, because red blood cells don't have a mitochondria, so they only use glucose as their primary or only energy source for that matter. And the brain does have mitochondria. It has a ton of mitochondria, actually, but it doesn't do beta oxidation because it uses glucose as its primary energy source for a few reasons. Beta oxidation doesn't produce enough oxygen for the brain. It generates oxidative stress, which you don't want in the brain. And ATP generation is slow in beta ox compared to like glycolysis. So the brain's mostly using glucose as energy, but in starvation we'll see that the brain uses ketones too. Some things that inhibit this pathway are, so this is a catabolism pathway. We are doing this pathway because we are in fasting or starvation, and we need to use alternative energy sources other than glucose for energy. So we're using our fatty acids. So insulin will inhibit this pathway because insulin is a hormone that stimulates synthesis pathways, and it signifies that you're in the fed state, and that's not the state we're in right now when we're breaking down fatty acids into acetyl-CoA for energy. High acetyl-CoA will inhibit this pathway because typically whatever the product of the pathway is, if we have it, we don't need to go through this pathway to produce it. If we have it, that means that we have done what we needed to do, and we can do something else now. We don't need to keep breaking down fatty acids to get acetyl-CoA if we have a ton of it. High ATP will inhibit this pathway because high ATP signifies high energy, and we're doing this pathway because we have low energy. So high energy would inhibit this pathway. We don't need to be breaking down anything if we have high energy. Malonyl-CoA is a molecule in fatty acid synthesis. So fatty acid synthesis is the opposite pathway of beta oxidation. If we have malonyl-CoA, that'll inhibit CAT1, and that'll stop this pathway from happening because malonyl-CoA is on when we're synthesizing fatty acids, and this is fatty acid catabolism. Stimulators of this pathway are glucagon, epinephrine, and ACTH, and these all go together typically. These are high in fasting state and starvation when we need to break down fatty acids because we don't have glucose as energy anymore. And increased fatty acids will stimulate this pathway because fatty acids are the substrate that we're starting with here. So if we have fatty acids, we can do this pathway. I hope those made sense. There's a few clinical correlates that we learned about. So MCAD is medium chain acyl CoA dehydrogenase deficiency. It's talking about this enzyme here. If we don't have this enzyme, or if it's inhibited, we can't do beta oxidation. So if we can't break down fatty acids, then we'll have a buildup of fatty acids, specifically medium chain fatty acids. And if we have a buildup of fatty acids, we'll have low blood glucose levels because we ran out of glucose and now we're using our fatty acids. Carnitine deficiency is if we don't eat enough carnitine, or there's some reason why we don't have any. Carnitine is needed to get into the matrix. So if we can't get into the mitochondrial matrix, we can't do beta oxidation. And that'll lead to hypoglycemia because if we can't use our fatty acids, we're gonna try and use up all of our glucose. And we'll have low ketones. Because we're essentially going to use if we can't break down fatty acids, we got to find other sources for energy. So we'll use our ketones, we'll use glucose as energy, we'll use everything else because we can't break down our fatty acids. And there's a few peroxisomal disorders, Zellweger's and adrenoleukodystrophy. And both of these diseases just signify a lack of functional peroxisomes. And if our peroxisomes aren't functioning, as we talked about, our peroxisomes are what break down fatty acids that are longer than 22 carbons. If we can't do that, then we'll have a buildup of long-chain fatty acids in our tissues. I hope that clarified something. See you in the next video.